finished up with the neural processing, what I consider the retina and beyond. So we're going to get beyond to the primary visual cortex to V1 and talk about neural processing and receptive fields and so forth at that level. I want to repeat a little bit to put us where we were. So we were talking about the lateral geniculate nucleus and that it's in the thalamus. Its function and the function of these thalamic nuclei is to summarize information, control information, organize information so that we can find that information that we need, that is important, that we want to pay attention to. At the level of the lateral geniculate nucleus, we have a retinotopic map, which means that we are maintaining the mapping that is on the retina. So we're looking at visual space and we see that A is close to B and B is close to C and A is far from C. And again, that's the distal stimulus. We get this proximal stimulus hitting the receptors where that is inverted, flipped by the optics of the eye, so that at the retina, the A is down, the A that was up is down, the C that is down is up, but we have that same kind of relationship, right? We're keeping that spatial information about the visual space. So A is close to B, B is close to C, and A is farther from C. That's what's happening at the retina, and we're going to see this same kind of mapping or organization in the lateral geniculate nucleus. I will say I make these distinctions that are made in the field where we talk about a certain number of things. One, the receptive fields. We just went through the receptive fields of the retinal ganglion cells. They have a center surround receptive field. We're actually going to see that same kind of receptive field here in the lateral geniculate nucleus. That's what the neurons are responding to. That's what's causing their firing rates or changing their firing rates or influencing the rate at which they're firing. The pathway through the brain, we're going from the photoreceptors to the bipolar cells to the retinal ganglion cells within the eye. The optic nerve leaves the eye and it takes that information to the lateral geniculate nucleus, which sends that information to the visual cortex in the occipital lobe. That's really the pathway through the brain. The mapping or organization of the structure itself, what we're talking about is how is the lateral geniculate nucleus itself organized, and it's organized in this kind of retinotopic map. At these layers of the lateral geniculate nucleus, it has 6 layers, which is similar to what happens out in the cortex. Out in our cerebral cortex, we have 6 layers of neurons where they're going to be responding to a certain thing at a certain place if we go in perpendicular, and each of those layers in those kind of rows, they are each doing their own kind of job. Well, the same thing is happening here in the thalamus at the lateral geniculate nucleus. We have this retinotopic map. I'm adding a D to my A, B, C, and D here. And so A is close to B in visual space, A is close to B on the retina, and A is close to B in the lateral geniculate nucleus. Those layers now, 1, 4, and 6 are going to be information coming in from the contralateral eye. Then our 2, 3, and 5 are what's shown here in red. That's going to be information coming in from the ipsilateral eye, so the eye that's on the same side of the brain. And again, that gets shuffled together beautifully and amazingly such that the point A in space matches up between those 2 eyes, and in those separate layers, we have those points matching up. So corresponding retinal points are going to be within a particular column in the lateral geniculate nucleus. Other structure, the other subcortical structure that I'm going to talk about more briefly than I'd like to. I think the superior colliculus is really cool. It is taking in information not just from our eyes about space, but multimodal spatial coordination. So it's creating these spatiotopic maps based on not just that I see movement over there, or I see a bright color over there, but that I heard a noise over there, or I felt someone touch me on my left arm, and it's on the same side that I'm hearing a noise and seeing movement, right? So all of that information is going to be used in helping me decide where do I want to move my eyes, this kind of eye movement control, finding localizing things in space is what the superior colliculus is allowing for. And it's taking in information from all of our sensory modalities in order to do this. And the lateral geniculate nucleus is sending its information onto the striate cortex, or V1, or the primary visual cortex. It has, again, many names. Whatever I say, I'm always talking about that same area, primary visual cortex in the occipital lobe. And these neurons, the neurons in the occipital lobe, in the visual cortex, they have their own receptive fields. So far, we've been center surround, which basically means I'm responding to little dots. I've pulled apart all my visual information, or pounded it into dust, basically, into little tiny pieces and parts. And now we're putting those little dots together, that dust together, into things like lines of a certain orientation. We're still at features. So the neurons in the primary visual cortex have been called feature detectors because they are still just telling us about the features of the environment. We have simple cells that respond to a particular orientation of line, horizontal, vertical, some level of oblique, and different neurons are going to be responding to vertical. Different neurons are going to be responding to some level of oblique, right? Complex cells are going to be responding to a particular orientation of line. So let's just say I have a vertical line that I'm responding to, and it's going to be a vertical line moving in a particular direction. It doesn't respond to a still vertical line, and it doesn't respond to a vertical line that's moving in the wrong direction. It's going to respond to a vertical line that's moving in a particular direction. But this could be an oblique line moving up. So my particular complex cells responding to specific orientations, moving in a specific line, moving in a specific direction. End-stopped cells are sometimes called hypercomplex cells, but they're end-stopped cells is a great name because it's telling us where things are kind of stopping, right? So it's not just the orientation of the line and the direction of movement, but it tells me where that line stops. What is the length of that line in my visual field? And what is the angle? If there's an angle coming together, if it's a 90-degree angle or a 45-degree angle, that all of that, I'm going to have a specific, all of that has to be specific to me. I might be responding to a 45-degree angle of on a vertical line that's moving to the left. And it's going to be a certain length within my kind of aperture, that little tiny piece that I get to see as a neuron. I'm going to walk us through examples of each of one of those, and I'm going to start with the simple cortical cell. So again, these are feature detectors. I'm detecting features with these neurons. And this particular neuron is going to respond to a vertical line that's in a particular place in space, because remember, we had a retinotopic map in the lateral geniculate nucleus. We're actually going to maintain that mapping in the visual cortex. So if something's up to the right of your head, of your visual space, it's going to be down and to the left in the visual cortex. And if you can imagine, or if you want to put up your finger, there's a line up there as you're looking straight forward. That vertical line hitting in the right place is going to stimulate a certain neuron. It's going to hit that excitatory space that you're seeing over to the left. We have excitatory space in the center, and then these kind of side by side, we have inhibitory space around that. If I'm hitting right in that excitatory space on a vertical line, we're going to see the most impulses per second. That's what our graph is showing, right? That if I'm vertical, I have the most impulses per second. As I get off of that vertical just a little bit and get a little bit oblique, I still respond, right? Because I'm still hitting a lot of that excitatory space, but now I'm hitting less of it, and I'm hitting a little bit of inhibitory space. And so we respond less and less. That neuron responds less and less, the more that particular orientation of line is hitting that inhibitory space. So it's going to be inhibited when I'm being horizontal, because I'm hitting more inhibitory space than excitatory space. So this is a simple cortical cell. I am going to put the link. So that down there is measuring the receptive field of a simple cell in the visual cortex. That is Hubel and Wiesel doing this recording from a cell. I'm going to mention here that you're going to hear the neuron firing, and I usually play about 2.5 to 3 minutes of it. It's a like 11-minute video, which you can watch the whole thing if you want. It's kind of meditative, but I usually show about 2 to 3 minutes in class. Well, I usually show about 2 minutes 35 seconds, right around there, and then I have a specific point that I make. But so right around there is plenty to get the idea. And then, I'm going to do that at the end of this video. I'm going to continue on to talk about the complex cell. So a complex cell responding to a particular orientation of line. In this case, that is moving in a particular direction. And in this case, what this picture is showing is they were actually, when Hubel and Wiesel were doing this work, they were still using center surround kind of pictures, because they had been working with earlier processing, and now they're in the visual cortex, and they assumed they were still at this kind of center surround, and we had dots somehow that we were putting together. And then what happened one day was the slide was coming down, and the cat was already hooked up, eye anesthetized, everything was hooked up, so they were measuring from the neuron, from a specific neuron. That slide came down, and as that horizontal line moved down, David Hubel is quoted as saying in their Nobel Prize winning speech, the neuron went off like a machine gun. So they weren't getting very much neural response to their dots. And here they heard this neural response like a machine gun. That's what that neuron responded to. A horizontal line moving in a downward direction. I'm going to give another example, though, of our complex cell that's from the book. So we have a complex cell, and you can see that what this particular complex cell in the left responds the best to is what looks like an oblique line that is almost just a little bit off vertical, and when it's moving up and to the right. That's what those little lines mean, right? That's neural firing. When it moves down and to the left, silence, no response from the neuron. Up and to the right, it responds. And as we move up and down from that in the picture, we're getting farther and farther off, right? In each direction, we're moving away from that particular orientation of line, and the neuron doesn't respond as well. It responds the best to this particular orientation of line moving up and to the right. If we look over to the right side, there's our end stop cell, and part of what this is showing is that it's also, this 90-degree angle is important. This orientation of line is important. The fact that this one's moving up and to the left, that's what's important. So it doesn't respond so much if it's moving down and to the right. But the other place where we see there's a difference in the neural firing is I respond the best at that second place, right? That much overlap with my receptive field. That's the link that I want that to be. If it's shorter, so if you think of the dark space or the shadow is the receptive field, if that's shorter and it's not as long, I respond some but not as much. If it gets too long, I don't really respond that much. And if it gets way too long, I don't respond at all. It has to be this particular length with the 90-degree angle moving up and to the left at that orientation. So that's how specific those cells get. And if we end up talking about movement or perceiving motion, you'll see that we need that for understanding true motion. I'm going to again encourage you to go watch the video of Hubel and Wiesel working with listening to the neuron of a cat respond to simple features, to simple orientations of line. If we look at our receptive fields, now again, receptive fields are what is influencing the firing rate of a neuron, whether it's excitatory or inhibitory. How am I influencing the neuron? At the level of the retinal ganglion cells, we have center surround receptive fields. At the level of the lateral geniculate nucleus, we have center surround receptive fields. We've broken the visual world down into little tiny dots. We put those dots together into features at the level of the visual cortex, but we're still at simple, pure features where simple cells respond to just orientation of line, complex cells respond to a particular orientation of line moving in a specific direction, and end-stop cells respond to a particular orientation of line moving in a specific direction that's a particular specific length and has a specific angle.